In this video, we will discuss about this T cell characterization in different parabiosis condition. So, what is this parabiosis? Parabiosis means that two mice club together in a way that they will have the same blood flow circulations. So, they have used three different conditions for this parabiosis. First one is this isochronic between young and young mice. And young mice is like 4-month old, and the old mice is like 20-month old. And for these heterochronic conditions, they used young mice and aged mice, and for these isochronic aged conditions, they use old mice with old mice. And for 28 days, they keep them together, and afterwards they did the single cell RNA-seq analysis, and they are able to find out different populations of these immune cells based on the differential expressions of the genes in the cells. And they also characterize that which cells are expressed more, or which cells are expressed in which amount in different populations, and these are the relative expressions amount. Once they have this information, now they start comparing the results or the different types of T cells populations in different conditions. This young isochronic, and this young mice from the heterochronic, and the adult mice from the heterochronic, and the isochronic aged mice. Not aged, not adult, it is aged in this context. So they are able to see that this naïve CD4 plus T cells goes down as they move from young mice populations to heterochronic mice populations, and it goes down to a minimum in the adult mice populations. And similarly, they also see in the result for this CD8 plus T cells. It also goes down as they move from young to old. However, for this effector memory CD8 plus T cells, it goes up as they move from young to old conditions. And also there is this naïve CD8 plus T cells, it also goes up as they move from young to adult. And these things they have quantified in this panel E, where we can see that naïve CD8 plus T cells as it goes down. So this is naïve CD8 plus T cells. So this naïve CD8 plus T cells go down, and this is CM CD8 plus cells, it goes up. And also the effector memory CD8 plus cells, it also goes up. The naïve CD8 plus T cells, CM is central memory here. So naïve CD8 plus T cells, it goes down. It's quantitative analysis is shown here. And also this memory CD8 plus T cells, or the CM CD8 plus T cells, it goes up as it is shown in the panel D, and the quantifications are shown in this panel E. Afterwards, for these things they have identified on overall T cell populations that they have collected from the specific site. And in the panel F, they have separated the cells by origin. That is, CD45.1 belongs to the young cells, and CD45.2 belongs to the aged cells. And based on this, the trend also follows the same things that they have seen in the panel D. However, the difference is further exaggerated. That is, this naïve CD4 plus T cells is significantly higher in the young populations compared to the populations in the aged mice, or in the aged cells, not aged mice, it's all in the aged cells. And similarly, this naïve CD8 plus T cells is also high, whereas it is significantly lower in the aged cells. And this effector memory T cells is almost negligible in the young cells, whereas significantly higher in the old cells. And this central memory CD8 plus T cells is also low in the young cell and significantly higher in the aged cells. And this effector memory CD8 plus T cells, based on the origin of the cells, is also shown here, which validate the finding observations in the panel F. That is, aged CD8 plus T cells are significantly higher, the effector memory CD plus T cells, compared to the young CD45 T cells. So that's all for this video. From this, they just wanted to make a point that the T cells composition changes based on the age CD8 plus T cells, or based on the young mice or aged mice blood flow circulations, or based on the presence of aged mice or young mice blood flow or immune cells. That's all for this video. If you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. Thank you for your time.